Hello, everyone. Good afternoon. My name is Jill Farner, and I am going to moderate this session. So this is something that was the brainchild of the Wiedemann Steiner Syndrome Foundation, and it's going to be a fireside chat. And so I'm going to moderate and ask questions. There's the fire. I'm going to moderate and ask questions of Dr. Hans Bjornsson and Dr. Wendy Jones. And these questions were submitted by members of the community and I believe voted on. And so the top questions were chosen, and I'm going to ask each of them these questions, and then we may have time at the end for some additional questions. So I want to start by introducing all of the speakers. So first of all, as I said, my name is Jill Farner. I direct the Epigenetics and Chromatin Clinic at Johns Hopkins in Baltimore. I'm an associate professor in the Department of Genetic Medicine at Johns Hopkins and also in the Department of Pediatrics. And my lab works on identifying models and therapies for disorders similar to Wiedemann Steiner Syndrome. So first, let me start by introducing Dr. Wendy Jones. Dr. Jones is a consultant in clinical genetics and genomic medicine at Great Ormond Street Hospital in London and an honorary associate professor at University College London in the Institute of Child Health. She has a specialist interest in chromatin disorders, including Wiedemann Steiner Syndrome, dysmorphology, genetic epilepsies, and disorders associated with hypertrichosis or increased hair growth. Dr. Jones jointly chairs the UK National Dysmorphology Meeting and is chair of the UK Clinical Genetics Cleft Clinical Excellence Network. She received her MBBS from University College London. After completing core medical training in London, she undertook training in clinical genetics at St. George's Hospital and Great Ormond Street Hospital for Children, both in London. During her clinical genetics training, she won a place on the Wellcome Trust PhD program for clinicians and was awarded a PhD by the University of Cambridge. Based on the Wellcome Trust Sanger Institute, she researched into Wiedemann Steiner Syndrome and disorders associated with hypertrichosis. In addition, she carried out some of the core genomic analyses for the Deciphering Developmental Disorders study, a groundbreaking UK-wide study with looking into the genetic cause of developmental disorders. And Dr. Hans Bjornsson is a professor of translational medicine and pediatrics at the University of Iceland, chief of the Department of Genetics and Molecular Biology at Landsbotali University Hospital, and associate professor of genetics and pediatrics part-time at the Johns Hopkins University. Dr. Bjornsson joined the faculty at the McKusick-Nathans Institute of Genetic Medicine in the Department of Pediatrics in 2012, where he founded the Epigenetics and Chromatin Clinic that I referred to before. His research has focused on how genetics, environment, and therapeutics modify epigenetics and disease states. And he hosted the first Wiedemann Steiner conference in 2015, which Wendy and I were also there, and has been actively researching Wiedemann Steiner syndrome for the last 3 years. And he's the mentor of Dr. Reynesdottir. Dr. Bjornsson has received numerous awards for both clinical care and scientific work, including the NIH Director's Early Independence Award in 2013, the William K. Bowes Jr. Award in Medical Genetics in 2014, and awards for exceptional contribution to Kabuki Syndrome research and care in 2016 and 2023. So now that I've introduced our speakers, thank you. We will just jump right into the questions. So first question. So we recognize the patient community is global and wanted to start off with a question on the population size. In your opinions, what is the population of those diagnosed with Wiedemann Steiner syndrome?
Brilliant. No, thanks, Jill. I'm happy to answer that one. So it's a pleasure to be here today. Thanks so much for the invitation to join you at this conference. So in terms of population diagnosed, I think there's probably two parts to this question. One is how many people in the world have Wiedemann Steiner syndrome, and then the other one is how many have been diagnosed, and those two figures are not the same. So we did some calculations based on how often you'd expect to have an alteration or mutation or change in the KMT2A gene, looking at the DNA, because obviously it's a change in a gene that causes Wiedemann Steiner syndrome. And you can look at the DNA and look at how often you'd expect there to be a new change in the DNA. And we've all got new changes in our genes, we all have, but obviously the change in the KMT2A gene that individuals with Wiedemann Steiner have, the KMT2A gene is an important gene, and that's why you have the features of the condition. So like using maths and looking at how often you'd expect to have a change in KMT2A as a new change, 1 in 25,000 to 1 in 40,000 people will have a significant change in the KMT2A gene and have Wiedemann Steiner syndrome. So if you look at the population of the world being about 8 billion, we'd expect in the world there'll be 200 to 300,000 people with Wiedemann Steiner syndrome. So that's about sort of just less than half the population of Denver. So there's about 700,000 people here. If you imagine that's just under half of those people in the world would have Wiedemann Steiner syndrome. But as you can imagine, many people won't be diagnosed. So across Africa, the instance will be the same, but people just don't access genetic testing. It isn't available. So the vast majority of people with Wiedemann Steiner syndrome won't know they have it. And in countries where there is genetic testing, many people who are adults might have had genetic testing when they were younger, but it's come on significantly, and so there'll be large numbers of adults who will have Wiedemann Steiner syndrome, but won't necessarily know that because they won't have had genetic testing. So about 200 to 300,000 people worldwide probably have it, but won't have had a diagnosis. In terms of how many people are diagnosed, I think probably a couple of hundred potentially in the UK that I would potentially know about, but it's definitely an under kind of diagnosis. And I guess one point I wanted to make, sort of thinking about progress, is that the first step to a treatment is having a diagnosis. So I think that about being diagnosed with Wiedemann Steiner syndrome is a positive step in terms of knowing what the cause of a child or individual's difficulties are, and then sort of knowing what to expect and then thinking about the future. So I hope that's answered that question. I don't know if you want to add anything, Hans, or anything to that.
I just wanted to start by saying it's such a pleasure to be here and a privilege to be part of this group. So thank you for having me back. So I haven't done as nice of a math as Wendy has, but I came to the same conclusion because in Iceland we do a lot of sequencing, more than most places, because of this company called Decode Genetics, so very low threshold to diagnose people. And in the last 4 to 5 years, we have 3 diagnoses with Wiedemann Steiner. So I came to the prevalence of like 1 to 10 to 20,000, something like that. So it's not too far off. But it's much more common than what people originally thought. And it was also seen in the DDD program is that when you look at the top found diagnoses, and I was kind of waiting for you to say that, but one of them, KMT2A was pretty high up on that.
Yeah. So I think I also recruited quite a lot of individuals who I thought might have Wiedemann Steiner syndrome to the DDD. So I have skewed it a bit by recruiting lots of families. But yes, it is a high hitter. Even in our other studies, it's still a high hitter in terms of a diagnosis. Yeah.
So, and thank you all for participating in our fingerprint study. One of our ideas was that this might be a diagnostic aid because there are many regions of the world, even regions within the US, where the genetics expertise is missing. And, we've kind of found with Kabuki syndrome that we can separate their fingerprints from other fingerprints, and we wanted to see whether we could do the same for Wiedemann Steiner with kind of 2 goals. One, that it would be a diagnostic aid for some underserved areas, but also another way to kind of raise awareness of the disorder, because I think the more people that know about Wiedemann Steiner, the better for this community. So.
Great, thank you both. Okay, the next question is, what is the best way for these patients to access the research and treatment options happening currently? Hans, do you want to start?
So I think the foundation has done a great job of kind of keeping people in the loop. When they do a great job with the website and so forth. I think there's a handful of people that are pretty dedicated to Wiedemann Steiner. So another way, if you want to keep track of what people are doing, you can keep track of the publications that are coming out of the labs, or at least my lab has like a Twitter page where we try to share our findings in kind of relatively simple language so that it's not just limited to scientists. So I think those are two good ways of kind of keeping track of what options. But that I talked about, advocating and raising awareness for Wiedemann Steiner. Participating in research is another way that we together as a community can kind of move things forward. So thank you all for participating.
Yes. Sorry, the question was kind of how can people participate, or how,
What is the best way to participate in research and treatment?
Research and treatment options. Yeah, so I think obviously research is optional, and there's lots of different ways you can be involved. And WSS Foundation have got lots of information about how to take part. In the UK, we've obviously individuals who've come from Europe or from the UK also can take part in the kind of rare X research. And in the UK, we've got a study called GenRock, which is based in Bristol, but I'm working with them. So that's for individuals under 16. So there's lots of different studies available, but as Hans mentioned as well, that sort of just telling people about Wiedemann Steiner Syndrome or genetic disorders, all of these things contribute to kind of further understanding and increasing knowledge and everything like that.
Thank you both. And I'll just add also at our epigenetics and chromatin clinic at Johns Hopkins, I see all of the patients from a genetic standpoint, and Dr. Jackie Harris sees everyone. And from a neurology and neurodevelopmental standpoint, we do it all in one visit. And we always talk to our patients, and I know many of you, we've seen you in clinic, some of you in clinic, and we'll always be happy to keep you updated on research and how to get involved as well. All right, next question. So with the role of KMT2A in gene expression within the body, how important is it for research involving younger children and adults? What research exists within the various age groups? Start, Hans?
So it's kind of two parts to this question. One question was about what KMT2A does. And KMT2A is like a regulator of many other genes. So even though this is just one gene, it probably has pretty broad effects. So it helps many different cell types decide which genes to keep active or not active. So, and my thought has always been that even though KMT2A is misbehaving, if we could understand the most critical genes that it regulates, it might be a way to understand the disease and maybe develop therapies for that disease. So that's kind of the part about the gene. And then the other part of the question was about the age groups. So, I think any way, your kid is maybe not going to be able to participate in every given research study, but thinking about whether it's a good fit for your kid at any age group, I think is worth it. And like I said, participating, if your child is up for it, none of us want to torture the kids, but if they're willing to participate, and it's very helpful, and it's quite helpful at every age group. And so I think that's kind of my take.
Yeah, brilliant. And no, I just wanted to add, I guess, that Jackie and Rowena spoke a lot about rare X and the burden of illness survey. I guess in terms of looking forward to the future and thinking about clinical trials, so perhaps trialing treatments or medications, you always need a study endpoint. So you need to work out whether it's actually improved the situation or not. And there's definitely a move to having kind of sort of patient or family-centered endpoints. So in the olden days, researchers would say, oh, what do we want to be? What do we want to achieve with this treatment? What's going to get better? Whereas actually, I think people are realizing now that's actually the wrong question. The question is what do families want to get better? So what would be the endpoint for this clinical trial? What are the key issues for families that we want to improve for them, and asking them, what do you want us to improve? And so I think the Rare X Burden of Illness survey, for anyone to take part in that one, is not necessarily even involving the child. It's something for parents, but that would certainly give a good insight for us into what types of things are the biggest challenges for families. So I guess, as well as the kind of children and adults and sort of an actual research going on, if you didn't want to involve your child in research, there are some research that people could take part in, which would be really helpful going forward when thinking about sort of treatments and trials and things like that.
Great, thank you. The next question is, thinking about the future, we recognize gene therapy is becoming more of a promise to treat rare diseases within shorter timelines than ever before. For Wiedemann Steiner Syndrome, how close is gene therapy for a treatment option? And I might want to open that question up a little bit more broadly to say not only gene therapy, which is a very specific type of treatment for genetic diseases, but maybe other targeted therapies as well. And I'll let Hans speak to that first.
So I think it's a very exciting time. There's a lot of things happening with this CRISPR, right? You can modify things very well now, and there are these other ways. There's something called antisense oligos and many different strategies. The difficulty with these strategies, even though they work really well in kind of a cell dish, or we call it in vitro, is that it's still very difficult to target the right cells, to get this system into the neurons or get it into, and in Wiedemann Steiner, we don't even know exactly, we have some ideas, but we haven't proven like which cells are most important and so forth. So there's definitely some more research that has to be done. For the project that Tinna presented earlier in this conference, my idea with that project was if we could show that there was treatability in this disorder, that other groups would jump on it, because there's 6,000 disorders, and people are picking the ones, we just came from a lecture where they were talking about this. There's a limited number of disorders that get a lot of attention because people think there's some malleability or some treatability there. So if we could get it over the hump here so that people would say, oh, Wiedemann Steiner, there's some, malleability there, then people will bring their new treatments and test it there. And we're kind of halfway there. So we've done this, what Tinna achieved during her PhD, which is pretty amazing, is she was able to show it in utero, but we'd love to try to get more funding to test later dates. It was technically a little bit difficult, but I still think it would be really helpful for this disorder if we could show that. So, yeah.
The question is funding the only obstacle to that happening?
Yeah, so, it was not for a lack of try for Tinna that we didn't do this already, because we started out, so I think the mouse is a clever model. It's kind of based on the stuff that I did with Kabuki for 10 years, and I wanted to do that in Kabuki, but I never got the money. And then when this opportunity came up, I was like, we should do this for Wiedemann Steiner. Because in some ways it's a much cleaner model than a drug model, because the drugs can have many different actions, but here we're just asking, hey, if we flip on the gene, does something get better? What doesn't get better? What does get better? And the difficulty was a practical difficulty. The mouse model that we used to kind of initiate the switch didn't work very well, at least not in our hands. But there are many such mouse models. And I think Tinna just kind of timed out there. So we ran out of funding and ran out of time with her. So so I think at least, we would have a good shot if we had some more money, but there's also some technical kind of hurdles. That being said, I also plan to share this model with the scientific community. When Tinna's paper comes out, the model is open to anyone. So if there's someone that knows how to do that better than we do, we're totally open for them to do that. So, and again, that's what, it's like this jumping on the bandwagon. Now that this model is available, I bet you there's going to be some research groups that are going to jump on it, and they have their strategy and they're good at something. But if we could get to a place where we had in early mice or something like that, to show that those also have some reversal, that would be a really big deal, I think. So that's my hope,
I'm going to ask one follow-up question to that, if that's okay, Hans. So I was thinking it might be helpful too, if you would be willing and want to share some more about your experience with Kabuki syndrome, you and Dr. Harris, and how you've sort of moved that along and where you are now, and how that might inform the timeline here, and possibly even some of the pathology, because the disorders are so closely related.
Yeah, it's been excruciatingly slow, I want to say, because for Kabuki syndrome, we have had some, basically we've had mouse evidence for 3 different therapies. One that could never go to patients because the therapy is a little toxic, but it was kind of like a proof of principle. And then we have 2 other therapies that are very well tolerated. And we were almost at a clinical trial a few years back. We spent some time with this company. It's a really big company. It's called Takeda. And then they decided that it was too rare of a disorder. So we had already kind of started to do some of these burden of disease studies and so forth. But the time frame there, I've been working on Kabuki for maybe, 15 years, close to 15 years. And we are now at the point where there are some clinical trials. There's one with the LSD1 inhibitor, which I really think will happen, and then Dr. Harris is working on a trial with something called the ketogenic diet that seemed to work really well in the mice. So those are 2 things that are moving to patients, which is exciting. But I'm very impatient, and I want it to happen quickly, and it's been too slow for me. I wanted things to move quicker. These 2 disorders, the genes are very, very similar. And even though there are differences, some of the aspects are very similar. Like they both have this visuospatial defect, which is pretty striking. And that's something that we found in the mice, but since has been replicated in the patients very well. So that's always nice when people can, you find something in a mouse, you're not sure if it replicates. The other thing that is pretty similar is the high sociability. I'm always struck by how much these kids want to interact with me and get to know me, and I really enjoy kind of interacting. But this is pretty similar to these 2 conditions. And my hope has always been also, if we can find kind of shared things between different conditions, then the group suddenly is much bigger. And then we don't have to face this issue where the drug companies say, oh, this is too rare for us to care about it or to work on it. So.
Thank you, Hans.
Well,
the question is Kabuki as rare as WSS?
Yeah,
So Kabuki, I think the figure people always gave was sort of 1 in 40,000. So they're probably about the same, I think. But Kabuki, I guess, was kind of more, for one reason or another, was sort of more known about. So people would make a diagnosis of Kabuki prior to us finding the gene. So I guess there's kind of more years of research underway for Kabuki. But I think the gene was discovered in 2010 for Kabuki. So there's only a couple of years between the gene discoveries. But people have been making a diagnosis of Kabuki for much longer, 10 or so years longer probably.
And just this jumping on the bandwagon, when I started working on Kabuki, I'm telling you, no one cared about the disorder. When I went to the patient family meetings, there had never been a physician that, for some of them, because there was no gene and people were very, would argue whether this was a real disorder or so forth. But it's been amazing to see that foundation grow. They have now a lot of money and a very organized and so forth. It's also been amazing to see this foundation grow and to see all these people and families attending. But they're a little bit ahead, further along, and there's a lot of people now working on that disorder, which is great, right? That's what we want for Wiedemann Steiner as well. So.
Thank you, Hans. Did you want to? Do you want me to repeat the original question?
No, it's fine. I think I was just going to add in terms of kind of gene therapies that I think there's been phenomenal progress in genetics, and it's it's really moving forward as a kind of a specialty. But I guess I feel excited about how many different treatment types there are available, and how things are being taken forward in other different conditions. And I think I'll just echo what Hans said, which is that Wiedemann Steiner syndrome is a genetic condition which is similar to other sort of similar genetic conditions. I think Jackie mentioned sort of sister or cousin disorders, but there's a sort of a group of disorders called chromatin disorders that the genes kind of function in similar ways. So the genes have roles in switching on and off other genes in the bodies. And so by knowing it's one of this sort of family of conditions is helpful, because actually any breakthroughs in the other conditions as well, and there's lots of scientists studying these conditions as a whole worldwide. In fact, even in the UK, we've got a chromatin network with tens of researchers and clinicians in it, which has just started up. But I think these types of networks and shared knowledge, give me kind of lots of encouragement that things are kind of moving forward and people are learning more. But as Hans said, we still have to try and make sure we understand everything about it. Some of these gene therapy treatments for other conditions, because they're certain that the brain is the area where the condition is being affected, involve sort of delivering the treatment into the brain through a catheter, into the spinal canal. So I guess we have to kind of be certain how things are going to be treated, or how things would be done, to know how it would be taken forward. But I think I'm excited by the fact that there's excellent researchers like Hans researching into Wiedemann Steiner syndrome, and there's also in the UK I'm working with some researchers in Oxford who again are sort of working thinking about mouse models of chromatin disorders, and they've got various other hypotheses involving the bowels and other things that might be going on. So I think there's lots of exciting research going on, and I feel positive about the future, that things are moving forward. But I appreciate to families everything seems quite slow, and there's sort of nothing immediately available. But I think we're in a really good position, poised going forward for advances in knowledge and treatments.
Thank you both. And I agree, and I'll just add, not only is the laboratory work important that Hans is doing and that what Wendy was referring to, but also Dr. Harris and Dr. Eng's work, getting clinical trial ready and getting studying and trying to really understand the neurological, neurobehavioral, cognitive features and how to measure those, so that when there is a therapy that we might be able to test, that there are actually outcome measures and biomarkers and things that we can actually follow to see if they're working. So all of that, I think, is very positive for Wiedemann Steiner syndrome and Kabuki syndrome, and so I think it's I think we're definitely moving forward, but I know it does feel slow for everyone. So, okay, moving on to the next question: what other promising options for research, like developing healthcare guidelines or drug repurposing, are there for Wiedemann Steiner syndrome? Do you want to go first, Wendy?
Yeah, so I'll talk about the management guidelines side of things. So very excitingly, there's around 21 kind of clinicians and psychologists meeting tomorrow from across the world. Some of us are face to face, and others will be joining us virtually. And we're putting together as a team sort of an international consensus statement for management guidelines. So practically, what does that mean? Is we're working together, looking at all the available proof or evidence in research papers, and using kind of clinical experience to write a document which is, I guess, when someone's been given a diagnosis of Wiedemann Steiner syndrome for them or a family member, what would be the best care they could get going forward, and looking at what things would be recommended for their medical care going forward. So this is involves reviewing the literature, discussing, and it's sort of a democratic process, so it will involve voting by the various experts to support guidelines going forward. And there are already kind of guidelines about, so practically what that might mean is suggesting you or your child has a heart scan, or suggesting that there's somebody coordinating your care going forward, or support at school and things like that. But this is kind of a document where it's very sort of evidence-based guideline going forward. And so this is something that, it's a standard of care for genetic conditions, and something that's not available for Wiedemann Steiner yet. In fact, there's many conditions it's not available for yet, but it's a really good step going forward in I guess, providing excellent care to individuals with WSS worldwide. So I'm really excited that we're doing that, and certainly we'll be sharing it will result in sort of a piece of paper, so a medical journal article that can be shared with people's doctors looking after them or their children going forward, but we'll be sharing that with the foundation, so they'll be able to direct everyone to that in the future as well.
Yeah, I just wanted to say I think the guidelines will be amazing, because I think it's going to be a lot of work, but I think it will, it's a tool that we can use to make the care more equal, so that people living anywhere can, if they get this diagnosis, their providers can follow those guidelines. So I think that's a major step. I just wanted to say, I think just like Jill said before, I think all the research is super important, because, like what Dr. Ng and Dr. Harris are finding are things that we can then use to say, hey, the stuff that we're looking in the mouse, is the relevant? Can we kind of pinpoint the most important kind of regions in the brain or so forth? So all of this research kind of builds together and kind of helps us kind of move things forward. So I think that's exciting, and thank you all for participating in it. So.
Great, thank you. The next question is, what are 1 or 2 actions that patients and families can do to help with research progress?
We've kind of touched on this before, but I think raising awareness is useful, because I think the more people, and maybe that's the difference between Kabuki and Wiedemann Steiner, I think a little bit more people know about Kabuki for whatever reasons. So awareness, fundraising, so, because sometimes even a little bit of money becomes more money, because, if a foundation like this funds something, like an example, like the money we received to support Tinna, she was also able to get a grant, so that money went a little bit further. Once you have like pilot data, you can then kind of build upon that. So, and then I think those are kind of like, and then participating in research. I think those are the 3 things that I would mention. So.
Yeah, no, similarly, I don't think I could add anything to that, but yeah, all of those things: awareness, taking part in research if you want to, and fundraising, those types of things, all good. Yeah.
Okay, the last question that was submitted is, what are you most excited about from a research standpoint for Wiedemann Steiner syndrome in the next 5 years? Let Wendy go first on this.
No, thank you. So I guess, I'm really excited about Jackie and Rowena's research. I think that we've got excellent people doing brilliant research in terms of understanding sort of kind of the neurocognitive difficulties individuals have, and understanding those. I guess ultimately everything we want to learn helps us both, kind of support individuals and give information to kind of health education providers and healthcare providers. And so I guess at this point, in terms of understanding the challenges or the differences individuals with WSS have, I think we're in a really good, exciting time where we're gaining lots of information. And I think and I guess, Jill also mentioned that, we really need this information to help individuals, but also to help show that, if there's a treatment, that things have got better. So I guess I'm, excited about everyone who's working on WSS, the kind of the climate we're in, where very quickly treatments are becoming available for different conditions, and so I feel it's a really exciting time. I feel we're kind of primed for things going forward. I guess for WSS, it's great to have Helen involved as well in the team. She's working with us on the management guidelines. So I think all of those things I feel very excited about going forward.
And I think it's a super exciting time, in genetics. There's, the speed is picking up. There are certain tools that are making us, like CRISPR being one, or AlphaFold, which is a way to predict how proteins fold. All these tools are kind of helping us speed up progress. And my hope in the next 5 years is that we'll have a better mechanistic understanding of Wiedemann Steiner. And maybe some, obviously I'm biased, but I hope with the mouse studies that we'll be able to show without a doubt that there's some of the aspects of the phenotype that can be treated. And I always say this, like people ask me, what's the window of opportunity? So like, when could you treat? And if the Kabuki stuff that we have done is correct, the answer is probably you can treat later in life, so whenever. But it's probably going to be more effective the earlier you start. So that's kind of like what we've learned from other diseases, and I think that's going to be hold true here as well. So.
Thank you, Hans. Thank you, Wendy. We can now open it up. Did you need to do anything about the streaming or the online before we open it up for questions? Or just make an announcement that it's still on?
Yeah, I think we'll go ahead and take the live stream down. We usually do that just for respect for